My name is Ferdia Gallagher. I am an academic radiologist from the University of Cambridge, and I'm talking today about hyperpolarized carbon-13 imaging in 2030, a clinician's view. This is a field that I've been working on for the last 15 years, and I'd like to describe to you today the transition into patients and some of the exciting clinical data that we now have in this field. These are my declarations of interest. To begin with, increasingly we are moving towards an era of multiscale imaging, shown on the left-hand side by the layers of imaging that we have available to us in radiology. At the very top is anatomical imaging, and the vast majority of clinical routine tools for imaging fall into this category. Increasingly, we can study physiology, blood flow, cellularity being examples of that. And below that, in the area of molecular imaging, we can look at metabolism. And there are a number of ways that we can do that with FDG PET, for example. Much of this talk will be centered on how we can use MRI to probe metabolism. And in layers below that, we can look at protein expression and even gene expression with imaging. Increasingly, we're moving towards an era of personalized medicine, where we can combine these tools in order to provide unique information on an individual patient. Do we need more imaging is an important question. This is just one very simple example of why that might be the case. On the left-hand side, we have a glioblastoma at baseline, and then following 3 months of therapy with temozolomide and radiotherapy, there's an apparent increase in the size of the tumour following gadolinium enhancement. But in surgery, this was demonstrated to have fibrinoid radiation necrosis and no tumour. And this concept of pseudoprogression occurs in somewhere between 15 and 30% of patients. And being able to have tools which can probe some of the more molecular characteristics of the tumour that I've just described on the left may help to reveal to us whether indeed the tumour is truly progressing or whether it is in fact responding to therapy, despite the fact that it's getting bigger. Another very key aspect for imaging is that it can look at tumour heterogeneity. And heterogeneity is key to most tumours. We know that more aggressive tumours tend to be heterogeneous. It's a major driver of therapy resistance, so understanding this may improve treatment response. It exists on many levels. It can be within a tumour, intratumoral, between tumours, intertumoral or intermetastatic, between patients, interpatient. It can be spatial or temporal. It can exist on a structural level, a functional level, or indeed a metabolic level, as I'll talk about today. And imaging is one of the key ways of identifying this. Although circulating biomarkers are increasingly finding their role in the way we manage patients, imaging gives us a true insight into the heterogeneity of a tumour and is very complementary to the information that can be provided by these circulating biomarkers, such as circulating tumour DNA. And this is an example of the heterogeneity that we can see on imaging in a patient with metastatic melanoma undergoing checkpoint inhibition. And as part of this trial, we've imaged the patients at baseline following 3 weeks of therapy and then following 12 to 14 weeks of therapy. And this is the work of one of our students in Cambridge, Doreen Lau. The middle histograms show the change in volume across 6 lesions in a single patient. And you can see there's a wide disparity in the response of these lesions. So some progressively get smaller across those 3 time points, some increase and then decrease, and some progressively increase. On the left-hand side, we can see the functional heterogeneity as measured with K-trans. As you'd expect, the majority of the lesions show a reduction in K-trans, post-cycle 1 and post-cycle 4. However, interestingly, lesion 3 shows a paradoxical increase at post-cycle 1. And then the top right demonstrates the interpatient heterogeneity across 6 patients with a wide variety of response patterns seen. So this is a nice example, I think, of how volume measurements alone are insufficient to measure response to therapy. And it shows examples of intratumoral, intermetastatic, and interpatient heterogeneity, all of which present challenges to imaging, and new methods will help us to evaluate this more accurately. So I've been asked to look to the future to see what hyperpolarized carbon MR will look like in 2030, which is a challenge. And in order to do that, I thought we could look back, in fact, and see what has changed in the last 10, 15 or more years and how that may relate to what we see in the future. So this is the very first paper published by Claes Goman, Jan Henrik Arden-Jacob Larsen and colleagues in PNAS in 2003. So this was the paper which initiated the field. Since 2003, there has been a significant increase in the number of publications that have been produced in this area, shown schematically here from a very simple PubMed search. And then this paper in 2013 was the first clinical paper which set the scene for the subsequent clinical studies that I will describe today. So the following are an example of some of the important papers that have been published in the clinical area since 2013. It's not an exhaustive list, and there are many other papers, but I thought it'd be nice to look at a spectrum of some of those which have made an important impact. For example, in 2016 through 2019, papers showing the first example of this technology in the human heart, the first example of it as a treatment response biomarker, and the first example in the normal human brain. And then on the right-hand side, this year we've seen a number of more clinical papers which have demonstrated applications in cancer such as breast cancer and prostate cancer, as well as applications in cardiac disease. So I'd like to begin by describing some of the setup that we have in Cambridge. On the left-hand side is the setup that we have in our 3 Tesla magnet. On the right-hand side, next to the PET MR, in each of those setups, we have a hatch through the wall which allows the passage of a hyperpolarized sample and injection into a patient. So key to all of this is the C13 pharmacy setup, and in Cambridge this is led by Matt Locke and his team. We have a dedicated room within radiopharmacy and a laminar flow hood in which the consumables for this, shown in the center of the image, the fluid paths are filled. The sample vial, shown on the left-hand side of the image, is filled with pyruvate. There's a coaxial tube that runs to a dissolution syringe which contains warmed water, and this passes down the center of the coaxial tube. It dissolves the pyruvate and passes back out through the coaxial tube, through a transfer tube and into the receiver vessel. The EPA filter removes the radical, the receiver vessel neutralizes the pyruvate, and the QC appendage is used with the QC module shown on the right-hand side in order to ensure it's safe for injection into the patient. In Cambridge, we fill in batches, we freeze our samples, and we use them as required, which is different to some of the sites in North America. So what will this look like in 2030? Well, first of all, the pharmacy kit or fluid path that I showed you on the previous slide will be simpler in design. There's already ongoing work to enable that to happen. What about centralized pharmacies? We have 4 sites in the UK which undertake this technology, 3 of which have their own pharmacies. And in the longer term, it may be appropriate to bring all of that together, such that one site would fill and send samples throughout the UK. And this simple model could also apply to sites in Europe and in North America. We've already demonstrated that it's feasible to transport within the UK and use a hyperpolarized pyruvate fluid path. Perhaps even more exciting would be transportable hyperpolarized molecules. And in this work by Arnaud Comot and colleagues, they've demonstrated very nicely how a frozen hyperpolarized solid material can be transported and used at a remote location at a later time point. So these advances will not only reduce the cost of the hyperpolarized technology, which is one of the challenges, one of the obstacles that has to be overcome, but also increase its reliability and availability across sites, particularly if we can transport hyperpolarized molecules. There's no need for a hyperpolarizer on site. We can decrease the need for specialist experience, which means that we can facilitate multi-site trials, which is really required in order to push this technology forward. And what about patient considerations? Well, we currently inject hyperpolarized pyruvate at a concentration of approximately 250 millimolar, the concentration at which it comes out of the spin lab. We give 0.4 mils per kilogram at a rate of 5 mils per second and follow this with a saline flush of 25 mils, again at 5 mils per second. The patient is set up for a breast patient is shown in the center, and on the right-hand side a selection of some of the coils that we use. Clamshell at the top, the breast coil in the middle, and a birdcage coil for brain imaging. In 2030, patient comfort will be a priority. We will have improved coil design to assist that, and it will be really key to integrate rapid imaging into the clinical pathway in order to make it feasible for routine patient imaging. And it's also important to have quantifiable information across large body areas. So one of the challenges with the current clamshell design is if we want to image large patients, for example, patients with intra-abdominal tumors. They may have ascites in the context of the tumor. Obesity is also a risk factor for many tumor types. And the current design restricts the anterior-posterior dimensions to about 28 centimeters and coverage in length of about 30 centimeters. Now, on top of that, the flip angles delivered may vary, as demonstrated with this phantom, and the coefficient of variation measured here was about 27%. Now that adds other problems in terms of quantifying across the abdomen, which is clearly an issue if we want to compare normal from abnormal tissue. Now, one potential solution to this is shown on the right-hand side. It's a prototype coil we've developed with Rapid. Josh Kagi will be presenting it at this ISMRM. And it has coverage which is now 50 centimeters in length compared to the previous 30. It's got excellent uniformity with a flip angle coefficient of variation of 16% and allows us, as you can see in the photograph, to image much larger subjects. The example sodium image is shown here, and it's very exquisite in terms of the detail that we can now identify on sodium MR. Now, that leads me to my next point, which is, can we use sodium imaging to inform on carbon imaging? Endogenous sodium signal is much more abundant than that for carbon-13, and yet their frequencies are very similar with a difference of less than 2 megahertz. We've previously shown that you can use the same coils to detect both nuclei in the prostate on the right-hand side. And in this work presented by James Grist at this year's ISMRM, we've shown that you can use the sodium frequency in order to set the transmit gain and the center frequency for carbon-13. So potentially we can build this into the clinical platform to improve our carbon-13 imaging. And moreover, we can get additional information as shown in the center imaging where we've fused sodium with carbon imaging. It's important not only to improve acquisition but also analysis methods, and in fact harmonizing analysis methods is key for future multi-center trials. And in this study by Charlie Daniels, looking at a phantom model with tubes containing different concentrations of enzymes, she compared these different approaches from, in the top left, the two-site exchange model, generating a KP or a KPL, which is an apparent exchange rate for the enzyme lactate dehydrogenase, through to more simple approaches looking at the graphical appearance, for example, the fall minus rise of the curves, area under the curve for both lactate and pyruvate, and time to peak lactate. In fact, these simple approaches correlated very closely with the modeled parameters. So potentially going forward, incorporating these relatively simple approaches may be key in order to incorporating them into clinical trials. So now I'd like to move on to the clinical applications of the future. Where do we see this technology being used in cancer to begin with? And the key reaction, which I've already alluded to in the previous slide, is the exchange of pyruvate to lactate under the action of lactate dehydrogenase with the exchange of NADH to NAD. The two major applications for this from the preclinical literature are firstly early treatment response, and this is the first paper demonstrating that in a lymphoma model within 24 hours of the treatment of etoposide, where there's a very significant reduction in the lactate over pyruvate ratio, as shown by the change from the bright yellow color to the deeper orange-red color. And the second is tumor grading from this paper on the right-hand side, which is shown in an example of prostate cancer, how going from normal prostate through low-grade and high-grade tumour, there's a progressive increase in lactate. And so much of my talk will centre on how we can use these two applications in the future. This is the seminal paper which showed it could be translated into patients and published in 2013, again in prostate cancer, and you can see within the peripheral zone on the left-hand side, in association with this area on the proton imaging, there's elevated lactate in keeping with the tumour there. And this particular case was very exciting because it demonstrated a tumour on the right peripheral zone, which showed elevated lactate as shown by the red voxels, but also there was no tumour visible in the proton MR within the left peripheral zone, but it also had elevated lactate, and they found a low-grade tumour when they went in and biopsied it. So potentially is metabolism revealing things which conventional proton imaging does not. This is the very first clinical example of treatment response, again in prostate cancer, from the UCSF group, showing a relatively large tumour on both T2 and diffusion-weighted imaging. There is some reduction in size in ADC following treatment, but a profound loss in metabolism as demonstrated in the KPL overlay before and after treatment. And that's only after 6 weeks of androgen ablation and chemotherapy. So potentially, can this be used in order to look for early treatment response? Can we build it into experimental trials? And can we use it as a clinical tool in the future? And those are some of the questions that we need to answer in the future. Moving on now to breast cancer. And this is the work of Ramona Wojtek in Cambridge, looking at different grades of breast cancer and how they may be phenotyped metabolically. Spectra are shown at the top, the dynamic contrast enhanced MR images in the middle, and some of the tumour characteristics below that. 5 tumours on the left are all grade 3 or higher grade tumours. 4 of those are the more aggressive triple negative tumours. All of those demonstrated visible lactate signal, whereas in the grade 2 hormone positive tumours, we didn't see any lactate present or very low levels of lactate. Now, importantly, that high lactate pyruvate correlates with larger tumours. We've shown in each individual tumour, we're using immunohistochemistry and RNA-seq that are related to the MCT1, as well as hypoxia in the form of HIF1 alpha. Now, these triple-negative tumours are the tumours most likely to undergo neoadjuvant therapy, and that's a key factor for us because it can be used to measure response to therapy. And this is the very first example in a patient with breast cancer, imaged with hyperpolarized pyruvate before and after neoadjuvant therapy. 1 cycle involved 3 weekly doses of Taxol and 1 dose of carboplatin. The hyperpolarized metabolism showed a decrease in both the lactate over pyruvate ratio and the KPL within that time. However, the DCE showed a very significant increase in the K-trans by more than 100%, and that increase in K-trans is normally associated with non-response. However, at the end of therapy, this patient had complete pathological response. So the hyperpolarized imaging is informing us in terms of the long-term response, and used in conjunction with proton imaging could potentially in the future more accurately predict response to therapy. So this may be one of the very key areas in which we use this technology in the future. Now I'd like to turn now towards brain metabolism, and this is the work of James Quist, published last year, looking at the normal brain and showing for the first time that we can image pyruvate lactin bicarbonate across an intact normal blood-brain barrier. We can see more pyruvate lactate and bicarbonate within grey matter compared to white matter. One of the questions we've been asking is, where does this lactate arise from? And potentially it's actually arising from the astrocytes, where pyruvate is taken up across the blood-brain barrier into the adjoining astrocytes, where the isoform of lactate dehydrogenase converts it into lactate. That lactate is shuttled across to the neurons, where a different isoform of LDH, LDH1, converts it back into pyruvate and ultimately into glutamate. So in fact, we may be looking at lactate arising from the astrocytes and potentially producing pyruvate that has been consumed by the neurons. We were able to quantify this now, and that's important for ongoing studies, so that we know that the regional variations are not only between white and grey matter, but was also between other areas of the brain where there are regional variations. And in the future, this could potentially be a very powerful tool for looking at disease processes where there's altered metabolism, albeit in the context of an attacked blood-brain barrier. A number of groups have been looking at the metabolism of pyruvate within brain tumours. These are two early examples from, on the left-hand side, UCSF, and on the right, Memorial Sloan Kettering. These studies have been assessing the formation of bicarbonate and lactate within the normal appearing brain parenchyma and a variety of tumours. A question that we have been trying to address in Cambridge is being able to phenotype glioblastoma using the hyperpolarizer. This is work of Fulvius Cagna, who's been looking at brain tumours on both proton and hyperpolarized carbon imaging. And the histograms on the bottom left demonstrate the huge variety in appearances that we're seeing within these tumours. For example, the tumour highlighted in red on the left has a lower metabolism of lactate in terms of the normal appearing brain parenchyma. And we use the term normal appearing brain parenchyma because although in proton MR it may appear normal, histologically we know these are very invasive tumours and extend well beyond the anatomical margins that we can see on proton imaging. Now, in contrast, on the tumour highlighted in red on the right, we see an elevation in the lactate over pyruvate in the tumour compared to the normal appearing brain parenchyma. And in fact, on average, the glycolytic metabolism within these glioblastomas is very similar between the normal appearing brain parenchyma and the tumours, and that's shown in the top right. We've been studying the biological drivers behind this using samples derived at surgery and shown that it correlates with LDHA expression in Western blot, but interestingly not volume. So this underlies the differences between tumours, whereas MCT1 may be the driver in prostate cancer and breast cancer, as I showed previously. Volume may be important in breast cancer, but LDHA may be the driver in these glioblastomas. What was interesting from this work is that all the tumours showed a reduction in bicarbonate production compared to the normal appearing brain parenchyma, and that's shown in the top right. This heterogeneity extends across many tumours, and this is an example of metabolic heterogeneity within renal cell carcinoma. It's the work of Stefan Ersprong. And you can see on the left-hand side that this relatively large tumour has areas which have high pyruvate but low lactate, and equally low pyruvate and high lactate. So Stefan has been looking at samples derived intraoperatively and postoperatively and comparing the lactate measurements with those that we measure on imaging and showing that there's a good correlation between the intraoperative and postoperative samples and the lactate labeling. But as you'd expect, there's elevation in lactate postoperatively because of loss of perfusion. And using this information, we can derive regions from the tumour which show different metabolic characteristics. And going forward, this may be an important area in which we can phenotype tumours such as renal cell carcinoma and glioblastoma in order to predict their response to therapy better and indeed detect that response when they're given therapy. And that leads me on to potential clinical roles for hyperpolarized carbon-13 MRI. Examples include tumour metabolic phenotyping, examples that are demonstrated in breast cancer, assessing metabolic heterogeneity in glioblastoma, for example, in renal cancer. Can we stratify tumours based on the lactate labeling, looking at more aggressive tumours and less aggressive tumours non-invasively? And a really key area is this fourth element, early treatment response monitoring. I've already shown you examples in breast and in prostate, and I think this is going to be a key area for us as a community going forward in order to identify much earlier than using conventional imaging by assessing metabolic response to therapy. We're able to image longitudinally over time. Hyperpolarizer also gives us the opportunity to image patients who are at most risk of irradiation, for example, children. And I think the future is that we need larger studies, we need multi-site studies in order to show the validity of the technique and the applications for the future. The talk so far has focused on cancer. I wanted to show you a few slides of the use of the technology in the heart. We know that there are alterations in metabolism in many types of cardiac disease. Furthermore, these changes in cardiac metabolism may be a cause rather than as a result of cardiac disease. There's increasing data now to show that metabolites can be used therapeutically. For example, succinate has been shown to reduce ischemia reperfusion injury, which is caused by mitochondrial reactive oxygen species. We know that pyruvic dehydrogenase, the enzyme that produces carbon dioxide and found in the inner leaflet of the mitochondria, plays a key role in oxidative metabolism. It's inhibited by increasing acetyl coenzyme from beta oxidation of fatty acids, and it's a very sensitive marker of metabolic abnormalities. We also know that metabolic changes occur in both acute and chronic cardiac diseases. For example, ischemia reperfusion and cardiac viability are responsible for metabolic changes. And in chronic disease, it may have applications in coronary heart disease, diabetes, cardiac failure, cardiac hypertrophy, cardiac toxicity secondary to therapies, or other metabolic disorders. And these are three examples of applying hyperpolarized carbon-13 MRI to cardiac disease. In the top left, in ischemia, this is from Claes Goman and colleagues, in a pig model showing that following ischemia, although there may be normal perfusion, there's a reduction in flux through pyruvic dehydrogenase as shown by the bicarbonate map as a consequence of damage in the left anterior descending territory. In the bottom left, work from Chuck Cunningham and colleagues showing the first example in a normal healthy volunteer human heart and demonstrating pyruvate predominantly within the ventricles on the left. In the middle image, we see bicarbonate, which is largely within the myocardium, and on the right-hand side, lactate, which is both within the myocardium and also within the ventricles. And this opens up the possibility of applying this to human disease as well. And then very recently, work from Damien Tyler and colleagues in Oxford showing that the metabolic flux through pyruvic dehydrogenase is decreased in patients with type 2 diabetes mellitus, and that this flux increases after an oral glucose challenge. The future may also see molecules beyond C1 labeled pyruvate making their way into routine clinical practice. The first of those that's already been injected into a patient is C2 labeled pyruvate, which can probe the citric acid cycle. This has been undertaken in UCSF. An example is shown in the top right, where we can see not only the pyruvate that's been injected, but we can also see C5 labeled glutamate that's forming within the normal brain. So potentially that opens up the applications by probing brain metabolism in new ways using this probe. The list also covers some molecules which are likely to make it into human studies over the next 10 years. Urea, for example, for perfusion. In Cambridge, we've been working on fumarate as a marker for necrosis. The bottom right shows an example of this in a preclinical model where following treatment, the malate that's formed from that fumarate as a consequence of necrosis increases, nearly doubles, and could be a marker very specifically for necrosis. We've undertaken our toxicology studies and plan to inject this into patients within the next year. Acetate is another molecule for looking at fatty acid synthesis that may find a role. Bicarbonate could be used for measuring extracellular pH. And lactate itself could be injected to look at glycolysis, the advantage there being that it can be injected at much higher concentrations than pyruvate. So where will hyperpolarized carbon-13 MRI be in 2030? We expect it to be simple and robust. It will be available in large research centres, but perhaps some regional centres as well. It will be reproducible, quantifiable. By that stage, we'll have undertaken more biological and mechanistic validation. The applications include cancer, and I've stressed the two that I think will be of great importance, particularly stratifying tumours based on lactate labelling and aggressiveness, and as an early treatment response biomarker. In the cardiac arena, it'll be used for assessing myocardial dysfunction. In the brain, we can look at non-invasive measures of brain metabolism, particularly across an intact blood-brain barrier. I think key is that we will have to incorporate these into large clinical studies as metabolic endpoints. Potentially, we could be using it in some instances as a routine measure of metabolism. And we will see more and more of large multi-center trials to really show the applications of this in a widespread setting. And finally, thanks to the very diverse group of people from a wide range of backgrounds in Cambridge that have made this project possible. Thanks also to our funders, and thank you for listening.